Hantaviruses possess a highly specific ecological transmission cycle. Unlike arboviruses that rely on mosquitoes or ticks, hantaviruses depend exclusively on wild rodent and insectivore reservoirs for their environmental persistence, their amplification, and their continued survival. Humans are incidental, dead-end hosts. Spillover occurs predominantly through the inhalation of aerosolized viral particles, continuously shed in the urine, feces, and saliva of infected rodents. Once inside a human host, the clinical pathology is severe. In the Americas, infection frequently precipitates hantavirus cardiopulmonary syndrome, or HCPS, an acute respiratory disease characterized by massive plasma leakage into the lungs, carrying a case fatality rate between 20 and 40%. In North America, the primary reservoir for the sin nombre hantavirus is the deer mouse, Paramiscus maniculatus. It is a ubiquitous keystone species vital to the continent's prairie and forest ecosystems. The historical approach to mitigating hantavirus risk has relied almost entirely on rudimentary environmental controls, primarily the physical extermination of rodent populations using chemical rodenticides like difascinone. This strategy is ecologically disruptive and ineffective in wild, open biomes. Localized culling operations are quickly neutralized by rapid rodent population rebounds and the continuous geographic influx of infected individuals from adjacent, untreated territories. High human lethality, the total absence of licensed human vaccines, and the persistent failure of lethal ecological controls necessitate a transition from broad extermination to targeted biological intervention. The defining biological hallmark of a hantavirus infection is its profoundly divergent outcome depending on the host species. In humans, the virus triggers massive immune dysregulation, a catastrophic cytokine storm that destroys endothelial barriers. In the deer mouse, the infection is lifelong but completely asymptomatic, lacking any cellular damage or behavioral impairment. This asymptomatic state is not the result of a failed immune response. The deer mouse immune system actively detects the virus and mounts a response. The lack of disease is driven by an evolved, actively regulated peripheral tolerance that restrains viral clearance to prevent host tissue damage. This chart contrasts the immune responses. In the human host, pro-inflammatory cytokines spike uncontrollably, but in deer mice, transforming growth factor beta follows a tightly regulated biphasic PERV. This cytokine suppresses the immune response, preventing vascular damage while allowing the virus to persist. To test if deer mice are capable of clearing the virus, researchers utilized a heterological infection model. They inoculated North American deer mice with the Andes virus, a hantavirus native to South America whose natural host is the long-tailed pygmy rice rat. Instead of establishing a persistent infection, the deer mice mounted a massive cellular immune response, actively hunting down the pathogen. Within 56 days, they completely cleared the Andes virus RNA from their systems, exhibiting zero clinical pathology. The deer mouse immune system possesses the inherent mechanical capacity to hunt down and neutralize a hantavirus without triggering fatal vascular leakage. Viral persistence is a specific evasion strategy that can be bypassed through artificial intervention. Developing precise therapeutics requires mapping the exact molecular mechanism the virus exploits to breach the host's cellular boundaries. This diagram shows protocoherin-1, or PCDH1, the obligate entry receptor for New World hantaviruses. The viral glycoprotein complex binds exclusively to its first extracellular domain, EC1. This interface is highly sensitive. Specific mutations, like F83L, disrupt this binding, physically preventing the virus from entering the cell. Mapping the PCDH1 EC1 domain identifies a highly specific molecular vulnerability. We now possess the exact biological target required to engineer host populations that are structurally resistant to infection. The first tier of the eradication framework relies on prophylactic immunization. To bypass the logistical barriers of vaccinating wild rodents, we utilize self-disseminating recombinant cytomegalovirus, or PCMV vectors. This abstract network map illustrates the mechanics of a self-disseminating vaccine. We administer the engineered vector carrying hantavirus antigens to a single founder individual. Through natural interactions like mating and allogrooming, the protective vaccine spreads through the population, magnifying the impact of a single inoculation. Cytomegalovirus is the optimal delivery vehicle because it is biologically constrained by host specificity. A CMV vector adapted exclusively for the deer mouse cannot cross the species barrier to infect humans, predators, or any non-target organisms in the ecosystem. Additionally, CMV induces a continuous proliferation of virus-specific CD8 plus T cells, a process called memory inflation. It also possesses a unique capacity for superinfection, allowing it to successfully immunize hosts that already carry pre-existing immunity to naturally circulating wild-type CMV strains. To ensure the autonomous vector outpaces the endemic virus, epidemiological SIR modeling calculates the exact mathematical insertion point into the fluctuating population. The release is mathematically optimized immediately following the peak of seasonal reproduction. At this precise juncture, the ecosystem holds the maximum number of immunologically naive juvenile mice, providing the vector with a highly susceptible, localized transmission network. Synchronizing PCMV's biological properties with the reservoir's breeding cycles creates self-sustaining herd immunity, capable of starving the hantavirus out of open biomes. While PCMV prevents future infections, treating an already infected persistent reservoir requires targeted therapeutics. The second tier of the intervention utilizes RNA interference, or RNAi. RNAi operates by introducing small interfering RNAs into the cell. These molecules hijack the host's endogenous RISC machinery, using it as a guide to locate and catalytically degrade conserved segments of the viral genome, completely arresting replication. The challenge is delivery. Naked double-stranded RNA is rapidly destroyed by endogenous nucleases in the bloodstream. Even if it reaches a cell, it becomes trapped and degraded within the cellular endosome before it can execute its therapeutic function. This illustration demonstrates the state-of-the-art lipid nanoparticles, the non-viral carriers engineered to solve this delivery failure. The therapeutic RNA is encapsulated within a complex, intact lipid shell that masks it from immune recognition. The shell relies on ionizable cationic lipids. These lipids remain neutral in the physiological pH of the bloodstream. However, as the nanoparticle is engulfed into the acidic environment of the maturing cellular endosome, the lipids protonate and acquire a strong positive charge. This sudden charge shift triggers an electrostatic rupture, tearing open the endosomal membrane. This mechanism, known as endosomal escape, safely deposits the siRNA payload directly into the cytoplasm, where it can dismantle the virus. We deploy these advanced nanocarriers into wild environments by embedding them into palatable, environmentally stable oral oat-groat bait matrices, bypassing the need to capture or inject individual rodents. When the deer mouse consumes the bait, specialized ligands coating the nanoparticles bind to receptors on the gut epithelium. This initiates active transcytosis, pulling the nanocarriers across the intestinal barrier and into systemic circulation. Advanced nanocarriers navigate the complete biological delivery pathway from the forest floor to the cytoplasm. A single oral dose can now execute a systemic cure within a chronically infected host. The third tier of intervention establishes genetic immunity using synthetic CRISPR-Cas9 gene drives. This pedigree chart compares standard Mendelian inheritance against a gene drive. Under natural conditions, a protective mutation only has a 50% chance of being passed down, often disappearing through genetic drift. A CRISPR gene drive overrides this math, actively copying itself into the genome of every offspring. The engineering tar date requires us to embed both the desired mutation, specifically the F83L alteration that blocks viral entry, and the Cas9 endonuclease machinery directly into the organism's germline DNA. When a modified deer mouse mates with a wild-type mouse, the Cas9 enzyme encoded by the drive is transcribed. It executes a precise double-strand break on the wild-type PCDH1 allele. To fix the break, the cell's endogenous homology-directed repair pathway uses the CRISPR-bearing chromosome as the blueprint. It repairs the DNA by copying the resistance mutation, converting a heterozygous embryo into a homozygous mutant. Over a brief span of generations, this results in the rapid exponential introgression of the uninfectable PCDH1 mutation through the entire localized ecosystem. This continuous copy and paste mechanism permanently transforms the keystone species into a structurally resistant host. Deprived of its obligate entry receptor, the pathogen faces environmental extinction. Actualizing autonomous biological interventions blurs the boundaries between localized veterinary medicine, public health, and macro-level ecosystem engineering. The Canadian regulatory framework serves as a template for managing these technologies. Under this system, recombinant vectors and genetically modified mammals carrying CRISPR gene drives are classified as animate products of biotechnology. The environmental release of these entities is governed by the statutory authority of the Canadian Environmental Protection Act, or CEPA, and the New Substances Notification Regulations. Before field deployment, sponsors must submit to an independent 120-day scientific review. Epidemiologists and geneticists map the risks of off-target genetic effects and horizontal gene transfer to non-target species. The exponential speed of synthetic biology requires rigorous oversight. Every intervention must undergo an independent statutory review to ensure that altering the genetics of a keystone species eradicates the virus while maintaining the broader ecological equilibrium. We no longer have to rely on the ecologically disruptive extermination of wild animals to manage zoonotic threats. By integrating autonomous PCMV vectors, RNAi nanocarriers, and targeted CRISPR gene drives, we can sever the hantavirus transmission chain directly at its ecological source. This approach illustrates the One Health framework, the reality that human public health, veterinary medicine, and ecosystem vitality are intrinsically linked. Instead of managing outbreaks after they occur, engineering resistance in the reservoir targets the virus before it reaches human populations. Targeted biotechnology transforms the reservoir from a spillover into a